Hello, I am Dongwook Kim from Seoul National University, South Korea, and I am glad to present my project in ISMB. Let me introduce UFCG, the database of universal fungal core genes and pipeline for genome-wide fungal phylogeny. UFCG provides all-in-one solution for fungal genomic phylogeny. Think about a mycologist here who wants to perform a fungal phylogenetic study. He or she might have questions such as which marker should I use, or what kind of tool should I use to draw a phylogenetic tree, or how can I do it fast? UFCG provides answer to these questions by providing a curated set of core genes associated with fully automated pipeline, which is scalable and flexible. We prepared a database that contains novel set of fungal core genes. We started from the resources publicly available. From the reviewed protein sequence database of SwissProt, we defined 41 novel core genes that covers 95% of the fungal species as a single copy. I'll cover the detailed process in a minute. On top of this, from fungal literature, we added 20 genes that has been canonically accepted. These 61 genes were defined as a set of core genes, which was then compiled into a database with relevant sequences and hidden Markov models. For this analysis, we used 12,000 genome assemblies from GenBank, from which we extracted core genes we defined and stored them as a species database. To computationally derive universal and single copy core genes, we had to search between two huge databases of SwissProt and GenBank. This could be done by using MM6 profile search, which is a rapid and sensitive tool for sequence profile alignment. For this, we generated MM6 profiles from SwissProt proteins and searched them against GenBank assemblies to generate MM6 alignments. From these alignments, we could define 41 genes that are present in more than 95% of the species as a single copy. Core genes are then validated to exist in more than 95% as a single copy from the assemblies that we didn't use from the previous step. We developed a pipeline that utilizes the core genes we defined for phylogenetic analysis. The pipeline works in a fully automated fashion all the way from the sequences to phylogenetic tree. The pipeline has two main modules. Profile module accepts multiple types of biological sequences, including fungal genomes, transcriptomes, and proteomes, and extracts core gene profiles from the sequences. Tree module then collects these core gene profiles to align and concatenate the gene sequences, which is then used to infer a phylogenetic tree. I would like to emphasize three key advantages of UFCG, which are scalability, flexibility, and consistency. UFCG allows analysis scaled up to thousands of genomes. Tree shown here was built with almost 1,600 fungal genomes. Using the profile module of the pipeline, you can easily extract UFCG profiles out of them. The tree module then automatically collects the core gene sequences from the profiles and aligns them into MSAs. Again, tree module automatically infers the phylogenetic tree from the alignments. For my machine, the entire process took about 3 hours, which means that the phylogenetic analysis of this scale can be easily done within a couple of days using UFCG. UFCG also allows flexible analysis with combined sequence types of genomes, transcriptomes, and proteomes. Tree here was built with combined types of sequences from 3 species under the order of Eurotialis. Let's zoom in a bit. As you can see, the pipeline successfully clustered same species into a monophyletic group, regardless of their sequence types. UFCG produces consistent results with previously described methods. Tree on the left is a UFCG tree that I showed earlier, and the tree on the right is Busco tree built with 758 genes from OrthoDB fungal lineage, which was proposed about 2 years ago. Comparing these trees in subphylum level, you can see the results are mostly consistent aside from 2 pairs of clades. But considering these mismatches, we could find the papers that agrees with either topology, which means the true topology of these pairs are still in controversy. As a summary, UFCG provides a novel set of curated core gene for genome-scale fungal phylogeny. We also provide a powerful pipeline that allows scalable and flexible phylogenetic analysis. Our database and pipeline is publicly available at ufcg.steinegelab.com. I would like to appreciate the fellow contributors of this project and all of our wonderful lab members. I also acknowledge Seoul National University and NRF Korea by providing funding to this research. Thank you for listening.